So we are a small biotech company developing molecules produced by the human microbiome that we hope will fight neurodegeneration. So bacteria have co-evolved with humans, and actually they are very important for our health. In fact, many recent studies have shown that a disturbance of the microbiome can lead to brain diseases. Our lead molecule is a microbiome-derived molecule that is important for protein production, and we do believe that it can be beneficial in neurodegenerative disorders such as Parkinson's and Alzheimer's disease. I'm going to have a quick word about our company. So we are a small team, but very diverse. Our CEO, Sophie, is the founder of the Microbiome Foundation, and she's located in France. So is our program manager, Lucy. David Imke is our Chief Scientific Officer, and he spent the last 15 years as the head of neuroscience at Amgen before joining us, and he is located in LA. Now, the team that is here in New York is here at the bottom. We are actually located in the Johnson & Johnson incubator so J Labs in Tribeca. And this is where the internship will take place. So the team is composed of John, who is our VP of Business Development, myself, YJ, who is our research scientist, and will be joining, Helen will be joining the team very soon. Just want to say a quick word about our main program and our lead molecule, that is curin. So curin is actually a hypermodified nucleobase that resembles guanine that is here, but it is associated with a cyclopentadiol that you can see here. So curin is actually solely made by bacteria, and it is made by the bacteria in our gut, and it is absorbed by the GI tract. And then it can go anywhere in any organs of the body, but it can also cross the blood-brain barrier. And there is an important amount of this molecule in the brain. We actually synthesize this molecule, curin, so chemically, and we call it STL101, and this is our lead molecule. What we know about this compound curin is that it actually integrates in specific transfer RNA that are important for mRNA translation. And when curin is integrated in those tRNA, then the mRNA translation is optimal and there is a proper folding of the proteins. Now, when in conditions where curin is depleted, you have no longer its integration into transfer RNA, leading to misfolding proteins and aggregates. And aggregates are a hallmark of many neurodegenerative disorders, such as Parkinson's disease. Parkinson's disease is caused by the death of dopaminergic neurons in the substantia nigra, and it leads to an accumulation of this alpha-synuclein aggregates in Lewy bodies. And so because of this role of curin in mRNA translation regulation, we do really believe that it can have a therapeutic effect in Parkinson's disease and other neurodegenerative disorders. And in fact, we already have shown using in vitro model of Parkinson's and Alzheimer's disease that STL101 is neuroprotective and can reduce the alpha-synuclein aggregation. In fact, we published this work last year, and you can have a look if you want more details about it. But the goal of this internship is really to actually understand the mechanism of action of this neuroprotective role of STL101. So we want to, I don't know why, okay. We want to understand what is the link between curin, STL101, so the role in its mRNA regulation and the link with potentially preventing Parkinson's disease by have an effect on cell survival and a decrease in alpha-synuclein aggregation, for example. So it is really an internship that will be in the lab, working very closely with our scientists, and it will require some cell culture, RNA purification, RT-qPCR, and some microscopy to look at reactive oxygen species. So the ACE engine will be under my supervision, but as I said, we'll work very closely with YJ, our research scientist, and Helen, who is also joining us. If you want to know more about our company, I invite you to visit our website.